Gracious Lord God, thank you so much for the opportunity that we have to come before you. Thank you for your marvelous creation, for placing us in it. And thank you for giving us a Savior who has promised eternal life to us if we just believe and if we confess our sins to him. Thank you so much for all that you've done for us. Please give us wisdom and insight as we listen to our talk tonight about DNA. Please give Lester the words and wisdom that he needs to give this talk, and please bless our time together in our conversation. We pray these things in Jesus' name. Amen. Okay, so tonight, here's our agenda. We are very fortunate to have Lester Marr speaking to us tonight on amazing DNA. I'll give him an introduction in a few minutes, but meanwhile, I do have a few announcements to make. I'm allowing about 45 minutes for him, and I'm showing here that we'll have as much time as we want, I think, for Q&A and discussion. I'm assuming that we won't be questioning you, Lester, for a whole hour, but maybe more like a half hour, but we'll see how things go. And let's see, here's our plan for the next couple of months. I'm kind of excited that these next two meetings give us an opportunity to consider two sister organizations. The first one next month is a conversation between Hugh Ross and BioLogos President Deb Haarsma, who is the president of, as I say, the president of BioLogos. And there's a lot of similarities, but there's also some differences between RTB and BioLogos. And they have a very polite conversation between them, and I think you'll enjoy it. And it gives you some idea of what BioLogos is all about. And you'll be able to see what the differences are between the two organizations as well. Then in March, I just saw this webinar the other day. It's a conversation between Dr. Günther Beckley, who is now with the Discovery Institute, the Center for Research and Culture, which is basically the intelligent design group in Seattle. And he is having a conversation with Dr. Fuz Rana. And the title of this webinar, actually it's a webinar, called The Fossil Record in Christianity. So next month, you get an opportunity to learn a little bit of something about BioLogos, and the following month about the Discovery Institute and the similarities and differences between intelligent design and RTB and BioLogos. So anyway, I think that's going to be kind of fun. We haven't really gone beyond that in terms of our planning. I have got a couple of invitations out to some more live talkers. In case you're thinking we do nothing but videos, tonight is not the exception to the rule, I hope. We will be having more live presentations later on. The next thing that I do in these meetings is I try to highlight some of the books that RTB has recently released, and this time I'll just be including three. This first one, called Thinking About Evolution, 25 Questions Christians Want Answered, is jointly written by Dr. A.J. Roberts, Fuz Rana, Sue Dykes, and Mark Perez. This was released late last year, and I think it's really good. It gives a comprehensive overview of RTB's position on evolution from multiple different angles: scientific, theological, and philosophical. It is in a question and answer format and provides detailed answers to questions that many people would have, raised both by believers and unbelievers. And you can find this book and the other two, as well as many other resources that I've mentioned at previous meetings, and a whole host of other resources for sale on the reasons.org website. This is Ken Sample's just released new book, Christianity cross-examined? Is it rational, relevant, and good? And in this book, Ken steps into the courtroom where Christianity's on trial. He goes beyond merely presenting a systematic case for the Christian faith, diving into specific accusations against it, from the conventional, if God created everything, who created God, to why doesn't the Bible condemn slavery? Does Christianity stand up to cross-examination? You be the judge. And then the third book is Fuz Rana's latest book, which was released late in 2021, and it's called Fit for a Purpose, and it shows how the anthropic principle includes biochemistry. The anthropic principle is the idea that the universe appears to be finely tuned to make advanced life, especially human life, possible at least one location in the universe. And the only one that we really know about at this point is ours, that is the planet Earth. But anthropic principle means, for instance, that if the four primary forces, gravity, electromagnetism, the strong nuclear force, and the weak nuclear force, were even slightly stronger or weaker, especially in comparison with each other, any life, especially advanced life, would not be possible, not just on Earth, but anywhere in the universe. And there are many more examples spelled out in many of the Reasons to Believe books. And in this one, Fit for a Purpose, Fuz has expanded this idea to include biochemistry. He shows that life would not be possible without the special properties of water, hydrogen, carbon, and oxygen, proteins, nucleic acids, membranes, and more. And this month, in appreciation for your gift to RTB of any amount, RTB will send you a copy of Fit for a Purpose, or an audio version, or both if you so choose. And our speaker tonight is Lester Marr, who is the president of the Vancouver, British Columbia chapter. That's correct. He's in Canada as we speak. He's a trained RTB apologist, and his subject is amazing DNA. He's a semi-retired civil engineer who designs airports and highways. He's on the board of Reasons to Believe Canada, and he's spoken to schools and church groups about Christianity, as well as hosting monthly Q&A sessions for skeptics.
So this is a presentation that I done probably a couple of years ago, and I've kind of tweaked it here and there. I will make this presentation available to anyone who wants it, slides and pictures and all. As Kirby mentioned, I'm a civil engineer. I'm a STEM person, science, technology, engineering, and I first discovered RTB about 13 years ago when I read Hugh Ross's Crater in the Cosmos. And as I was going through university, I was kind of into young Earth, but when I started reading about Hugh Ross, it's like, wow, this makes a lot of sense. And then about 13 years ago, I attended a live lecture from Hugh. He was speaking on why the universe is the way it is. And after the lecture, I started peppering him with questions. You know, what about dinosaurs? What about Noah's Ark? What about this? And as he started providing answers, I was thinking, man, this makes a lot of sense. You know, where has RTB been, you know, when I became a Christian at university? So because I was so excited about RTB, I decided to join the organization. I later became president of the Vancouver chapter, and I'm also part of the board of directors for RTB Canada. So you're probably wondering, why is Lester talking about DNA? Because as an engineer, I'm more focused on math, science, math, physics, and chemistry. And to tell you the truth, I never took a biology course in high school, nor did I take any biology courses in university. In fact, the sight of blood makes me squeamish. My wife, who's a nurse, you know, shows me all these illustrations about operations, and my knees just go weak. I think that one of the benefits I have as a engineer talking about DNA is I can see the big picture. Now, don't get me wrong, I'm not a fuzz rana. I'm not going to go into the intricacies of DNA, but in terms of the big picture, I try to make it easy to understand. And like I said before, this PowerPoint is available, and you can, I will give you the link, and you can use it to share to your church groups and other people that you want to share this presentation to. I'd like to begin with what is a worldview? Now, it doesn't matter whether you're a Christian or an atheist, Muslim, Hindu, or Buddhist, your worldview has to answer 4 fundamental questions. And the first question your worldview has to answer is, what is the meaning of life? Essentially, why am I here? That's the first question. The second question your worldview has to answer is, where does morality come from? What is the source? Are there any absolutes, or is it relative based on society and the times? So that's the second question. The third question your worldview has to answer is destiny. What happens when people die? Is there a heaven, or are they just annihilated? And the fourth question has to do with origins. Your worldview has to answer, what is the origin of the universe, the origin of life, and the origin of consciousness? And luckily, our technology allows us to study the universe and DNA. So for the origin of the universe, we have the Hubble and now the James Webb satellite, which is just unfolding right now. But as well as electron microscopes allows us to see into the intricacies of DNA. And that's what we'll be looking at now, is the origin of life, and specifically DNA. So the big question is DNA a sum of material plus time plus chance, or is there an elegant designer behind it all? I'd like to start off from the Christian worldview perspective. And what we know from science is, in the early days of the Earth, after the great heavy bombardment, the surface of the Earth was just molten and it was unfit for life. Later on, as it cooled, we had water, but it was still sterile. And the Christian worldview in Genesis 1:2 gives an insight on what the origin of life came from. So we see in Genesis 1, it says, in the beginning, God created the heavens and the earth. Now the earth was formless and empty. Darkness was over the surface of the deep, and the Spirit of God was hovering over the waters. This word that we see, hovering, is a unique word. It's only shown twice in the entire Old Testament. And that's found in Deuteronomy 32:11, where it talks about the eagle or a vulture hovering over its young. So the Hebrew word for hovering is rakop. And according to Brown Drivers Briggs, the encyclopedia or the dictionary for Hebrew words, it says that rakop means hovering over the face of the waters, or perhaps a brooding and fertilizing. So I like to look at the Message and their translation, and it's by Eugene Peterson, and he translates Genesis 1:2 this way. It says, the earth was a soup of nothingness, a bottomless emptiness, an inky blackness, and God's Spirit brooded like a bird above the watery abyss. So from a Christian perspective, the origin of life, as if the Holy Spirit brooded over the earth and created something from nothing. So that would be the RTB perspective in terms of the origin of life, and that kind of addresses the worldview question. Today's lecture will be going through this outline, and first we'll be talking about DNA. Is it an information or a pattern? We'll be looking at the basic structure. We'll look at redundancy, optimization, homochirality, data storage, and a process of going from DNA transcribing to RNA and then transcripting to translating to hemoglobin. So let's look at information versus pattern. When we look at information, information has two forms. One is a symbolic form, which is symbolic information, and then there's the implementation of it. So here we see music in its symbolic form. It is a series of notes, and when you apply and implement these notes, you come up with a song. So I just happened to meet Elvis in Las Vegas one day, so I decided to take a picture with him. Another form of information would be binary code. So in this case, we have a series of ones and zeros, which is symbolic. But when we implement this binary code, we come up with Microsoft Windows or Apple OS or whatever program that you have. So this symbolic information can be implemented into a computer program. So the question is, when we look at music and this type of information, information is based on a language, it has exact copies, and it requires intelligent thought. However, when we look at a pattern, for example, snowflakes come from chaos. They have no exact copies, and there is no intelligent thought required. So the big question when we talk about DNA and we look at it from a big picture perspective, is DNA information or is it a pattern? Is DNA a language or is it chaos? Do we see multiple copies of DNA or no exact copies? Do we see elegant thought or is it just simple matter that just happened to clump together? Next, we'll look at the DNA basic structure. This is a slide that that Richard Dean, who is a RTB apologist, allowed me to use. If we look at the DNA structure, we see that it looks like a twisted ladder, and the sides of the ladder is made up of a sugar phosphate backbone, and then connecting the two sides of the sugar phosphate backbone are nucleotide base pairs. And as we look at the nucleotide base pairs, we have thymine, adenine, and guanine and cytosine. What's interesting about the DNA is the thymine always pairs up with the adenine and the guanine always pairs up with the cytosine. So what that means is if you have one side of the DNA wrong, you will automatically get the other side because they are all matching base pairs. What's interesting about DNA is, like I said before, we have adenine, thymine, cytosine, and guanine. But when we talk about simple bacteria, it's not just a clump of jelly. In simple bacteria, scientists have discovered that you need at least 500,000 base pairs in sequence in order for simple bacteria to exist. So that makes it very difficult in the sense that if we have a primordial soup and life beginning, in order for simple bacteria just to exist, you have to have almost half a million base pairs just for simple bacteria to exist, let alone replicate or reproduce. When it comes to humans, the number of base pair increases from 500,000 to 3 billion base pairs. Now, one thing interesting and amazing about DNA strands is that it's highly coiled and wound up. Now, just from a show of hands, how many people think that if you were to uncoil a DNA strand, how long would it be? Would it be 1 nanometer? Maybe have a show of hands. How many think it's a nanometer? Or if it's 10 microns, maybe a show of hands. How many think it's 10 microns? Or 2 millimeters? Or is it 2 meters long? Well, Kirby knows his stuff. It is 2 meters long. So if you were to unwind and stretch a strand of DNA, it is 2 meters long. So here's another question. if you were to take all the DNA in your body and stretch it out and place it end to end, how long would it be? Would it be from San Jose to Paris and back? Or would it be from San Jose to the moon and back? Or would it be from San Jose to the sun and back? How many people think it's Paris? Okay. How many people think it's to the moon and back? Nobody. Okay. How many people think it's from the sun and back? Okay. It's actually neither. This was a trick question. It's not from the sun and back. It's from the sun and back 70 times, right? That's a long distance. So there's a lot of DNA in your body. That's 10 trillion cells at 2 meters long. That's yeah, it's just mind blowing when it comes to how much DNA is actually inside your body. What I'd like to do is I'd like to share a little bit I find that this video that I found from the DNA Learning Center on YouTube gives a short 2-minute clip, and it describes the DNA packaging as well as the replication, the transcription, as well as the translation into proteins. I will show 2 slides at a time. So we'll first look at how DNA is packed and how finely wound it is, as well as after that, we'll look at the replication of DNA. And this is how DNA is replicated. It splits the DNA in half, and then it forms the missing base pair in order to create another exact duplicate strand of DNA. Okay, so this is a YouTube video from the DNA Learning Center. We're going to look at the packaging of DNA. Let's put it on play.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
So next we'll look at DNA replication. So the chromosomes that we see, as they said, only occurs when you're about ready to divide. The DNA in this case is not always tightly wound, and that allows the mechanism for DNA replication to occur. As you look at this video, what's amazing is you'll see that not just the DNA, but there's machines that help put the DNA and replicate the DNA. So just think about it. You've got these micromolecular machines that are doing all this work. And for me to think that this came just out of random chance is just mind-blowing. So I will show you how this DNA strand is split in half and how the base pairs are rematched in order to create another identical strand of DNA. And because DNA is split, there's one side can be easily replicated, but the other side, you can't do it easily. You have to do a bit and then you do it at a section at a time backwards. This video will actually show you the process.
Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules.
Can you, am I back to PowerPoint?
Yes.
Yeah, okay. So, yeah, when I look at that, when I look at the DNA, the fine winding of DNA plus not just the DNA strand of 3 billion base pairs, but how it's replicated, to me, that's just simply amazing. Let's look at DNA redundancy. So a lot of you have worked with computers for a while, and what happens when Microsoft Windows or Apple OS gets corrupted? And if you're like me back in, I guess, the 1980s or 1990s, we were dealing with Windows 3.1, and if there was a corrupt bit in the code, what happens is you would get something called the blue screen of death. And essentially, as you're working along doing your engineering calculations, all of a sudden, boom, you get the blue screen, and then you start to curse, and you realize you need to reboot your machine because something went wrong with the computer code. Now, I'm not an Apple person, but from what I understand, the equivalent would be something they call the spinning beach ball of death. If you get this on your Apple Mac or your Apple machine, once you get the spinning beach ball of death, it's similar. Something has gone wrong with the computer code and you need to reboot your machine. So as we look at the computer code, we're looking at the genetic code. Before we talk about the genetic code, we have to talk about the DNA being transcribed to RNA. And the genetic code is related to the RNA. So what happens is DNA is copied to something called RNA, and it's almost, they follow the same base pairs, but instead of thymine in the DNA, it's replaced with a base pair called uracil in RNA. And the reason, you're thinking, well, why not use T in the RNA? And the reason being is the body is quite smart. It knows that thymine is very stable, but it takes a lot of energy to create the molecule thymine, whereas uracil is less stable, but that's not really an issue because RNA is short-lived. It only is used to create proteins, and once the RNA is finished, then it has a much shorter life. Whereas DNA, we want that to last for a much longer time. So as we look at DNA versus RNA, we see that both have the adenine, cytosine, and guanine. And the big difference is instead of instead of thymine, we have uracil. And if we look at the molecules for thymine and uracil, we'll see that one has a methyl molecule attached, whereas the uracil is much simpler. It just has a hydrogen atom. And on this corner here, we have the deoxyribose molecule, whereas on the uracil, it's much simpler and it just shows the ribose molecule. So from an energy point of view, it takes a lot less work to create uracil versus thymine. And by the way, I got this slide from Richard Dean. In fact, I've used quite a lot of slides from him. So what I'd like to do is I'd like to show you a little bit about DNA transcription. And this is how the DNA gets split off and how it's being able to match up and create a RNA strand from the DNA cell. So as it's trying to match the A's with the A's, the G's with the G's, the C with the C's, but when it comes to a T, it changes that to a uracil. And also, after we finish watching that video, we'll go to the next one where we look at the RNA and how it's further spliced using these molecular machines to create messenger RNA. And that messenger RNA is used to translate into proteins. Okay, so this is what we call DNA transcription. So it's transcribing from a DNA strand, one half of the DNA strand, and creating a strand of RNA.
What you are about to see is DNA's most extraordinary secret: how a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
So what that did was it took a strand or a section of DNA and it transcribed it into a section of RNA, but that RNA still needs some further cleanup to create messenger RNA. And we will look at some machinery of how the body splices the RNA to make it into a usable messenger RNA. And we'll be playing it now.
As DNA is transcribed into RNA, it needs to be edited to remove non-coding regions or introns shown in green. This editing process is called splicing, which involves removing the introns, leaving only the yellow protein coding regions called exons. RNA splicing begins with assembly of helper proteins at the intron-exon borders. These splicing factors act as beacons to guide small nuclear viboproteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes, shown here in purple, assemble along the RNA. Each spliceosome removes one intron, releasing the loop before disassembling. In this example, 3 introns are removed from the RNA to leave the complete instructions for a protein.
So that kind of explains that. So when you transcribe the DNA, you get a long strand of RNA, but as it has to go through splicing, and once you have after it's finished splicing, then you have something called messenger RNA, and that messenger RNA is used to code proteins from amino acids. So when we look at the genetic code, we see from the RNA that we have 4 letters. We have uracil, cytosine, adenine, and guanine. And the genetic code uses 3-letter combinations, which we call a codon. So if you look at these 4 letters, how many ways can you rearrange these 4 letters in 3-letter combinations? And if you're a statistician, or mathematically, there's 4 to the power of 3, which is 64 possible combinations or codons. These are the 3-letter codes. What makes the genetic code amazing is each 64 codon only codes for 20 amino acids, which means that there's redundancy in the codons. For instance, this is the genetic code. So for instance, if you have a letter uracil with a uracil as a second letter and a uracil as a third letter, you have UUU. So these are the 64 possible combinations for a codon. And what we see here is uracil adenine creates a amino acid called leucine. And we see here that it's not just one codon, but we have actually 8 possible combinations of codons, which will create, provide the same amino acid leucine. We have something special here called AUG, and that is called methionine. And MET is the genetic codon. It's the codon which tells the transcription to start. We also have 3 special codons, UAA, UAG, and UGA, and that is the stop. It tells the transcription to stop coding. We also have serine, which is located, which starts with a UC, but also has the AG prefix. And then we have arginine, which not just starts with the prefix CG, but also has the prefix AG. So this is what we call the genetic code. This is the basis of creating proteins from these amino acids, which these codons code for. So this kind of seems well, what does it really mean? I have a video which puts it all together. And this is what we call the messenger RNA translation. So this is where after the spliced RNA is finished, this RNA goes through another process where it takes 3-letter codes of the RNA and matches it to a specific amino acid. This is messenger RNA translation. So this is taking the RNA that's been spliced and cleaned up, and this will be used to go through these special molecules which will read each 3-letter codon and attach a amino acid to create a protein chain.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
So when it comes to optimization, back in 2000, they did a study where they randomly generated 1 million genetic codes, DNA codes, in terms of testing to see how the certain genetic code counters substitutional mutation. So if you get, actually, let me just go back 2 slides over. The nice thing about the genetic code is if you have, let's say for leucine, if you have CUU, and let's say you have a genetic defect and it reads CUC or CUA or CUG, what happens is it still creates the same leucine amino acid, which provides a lot of redundancy. So similar with arginine, you could have 1 letter off and it would still produce arginine. And even if you have a totally different letter, instead of CGA, you have AGA, it still produces arginine. So there is a redundancy within the genetic code. Now, even with that redundancy, what scientists have done is they've generated a 6 billion genetic code, and they've randomly did it, and they analyzed each individual code to see how it rates in terms of error minimization. So what they did is they took the 1 million randomly generated codes, and then they analyzed it, and what you have is you have your normal distribution. So this is the average performance for the 1 million random codes. On this side, you have the worst performing codes in terms of data error minimizing, but on this side here, we have the best of the best in terms of the least errors recorded if you have a substitutional mutation. Now, if you've taken statistics back in high school, you'll know that you can calculate the standard deviation. And if you were to take 3 standard deviations out, you're probably around this area. This represents 99.7% of all the best performing codes. So based on this normal distribution, if you're somewhere near this area, you're 0.3% better than any other code that exists. The interesting thing is they plugged in the human DNA code and they've wondered, where does this sit on the normal distribution? And instead of being on this distribution curve, they found that the human genome actually is somewhere out here. So it's not just 3 standard deviations, but it's 6 standard deviations from the mean, which means that this genetic code is 99.9999994% better than all these million codes combined. So I suppose if you were to take 1 trillion codes, then yes, that would probably show this on the if you were to increase this normal distribution with about a zillion codes, you would be close to where the human DNA code is in terms of substitutional mutations. And for me to say, or for someone to say that the human DNA is just due to random chance, yeah, maybe. If it's random chance, you'd be somewhere on this distribution. But the fact that the human DNA is 6 standard deviations from the norm says that there's something extremely special about our DNA. So when it comes to humans, we have 3 billion base pairs, and even if we have base pair errors, we don't suddenly freeze up as people. We don't get the blue screen of death or the spinning beach ball of death. We still are able to function. And to me, that's just amazing. Next, we're going to look at DNA optimization. This is a kind of a fun or interesting one. I have 3 sentences here. We have a man, a plan, a canal, Panama. The second sentence is live not on evil. Or the third sentence, was it a car or a cat that I saw? This is my sister's cat. So how many people recognize what makes these sentences very special? Maybe have a show of hands. Okay. These are not just random sentences, but we call these palindromes. And the reason being is, as you read this, a man, a plan, a canal, Panama, these can be read backwards. So a man, a plan, a canal, Panama, or live not on evil, right? And was that a cat or was that a car or cat I saw? So these sentences can be read forwards or backwards, and it means the same thing. And it takes quite a lot of intelligence to figure out a palindrome, but this is nothing compared to DNA. So what we find in DNA is there's different reading frames that you can apply to the genetic code. And these overlapping codes also give you the histone binding code, the regulatory protein binding, signal splice signaling, and RNA secondary structure code. It's actually best shown by another slide by Richard Deem, and he allowed me to show this to you. So this is half a strand of DNA. So you have the cytosine, thymine, guanine, cytosine. So as you read each 3-letter combination, you come up with a separate amino acid. So as you can see here, we're reading it in steps of 3. But the interesting thing is, this is what happens if you shift the reading frame over. Instead of starting with a CTG, you start with a TGC. Well, what happens is, instead of leucine, you have a different amino acid. And not only that, you have a totally different chain of proteins. And you can do this again. Let's shift the reading frame by another letter. And what you have is you have another different amino acid chain. But not only that, remember, this is only half the side of the DNA. You have the other side of the DNA chain. And once again, reading it from this direction, you have another set of letters where it creates another set of amino acid chains. But not only that, but if you shift this reading frame another letter, you have another different set of proteins. And if you shift it again, you have another set of proteins. So within this short section of DNA, it is possible to read 3 separate amino acid chains. And it actually works. Now, what's interesting is we have here something called the MET, and that's the start. It tells the ribosome, or when they transcribe it, tells this is where you start. And then here we have a code for stopping. So to me, that's just simply amazing in the sense that it's not that we just read DNA in one sequence. You can actually read it forwards and backwards, but not just only forwards and backwards, but you can read it 3 times going in one direction and 3 times in the other direction. And that is extreme fine coding. I mean, if some of you are programmers, you can imagine writing a program and then having subroutines. Well, in this case, you've got the subroutines built inside your main program. And that is incredible computer program. And to me, that's amazing. A long time ago, back in 1970, Francis Crick coined the term junk DNA. And that's because when they first studied DNA, they thought, well, hey, 97% of it doesn't have any apparent function. And so, they said this has got to be due to baggage, leftover baggage from evolution. And as a result of this attitude, it stopped the genomic research for decades. And it wasn't until the ENCODE project started in 2002, they published a report in Nature magazine. And what they found from the ENCODE project, after going through the analysis of sequencing DNA, they found that 80% of the genome is transcribed. So normally the body would not transcribe anything because obviously it uses energy. So they think that it's biochemically functional. And these non-coding DNA's bases act as switches and signal signposts. It also helps with gene regulation and expression. ENCODE 2 will kick up soon. And so it's likely that as we study DNA in a deeper way, we'll probably see that more and more of the DNA is functional. And to me, the fact that DNA is not junk and it seems to be doing something is amazing. Next, we'll look at homochirality. And homochirality has to do with the handedness of a molecule. So amino acids and sugars can be left-handed or right-handed. So it's kind of like your hands. You have 5 fingers and a thumb, but they're a mirror image of each other, but they're not exactly the same. And that's the same with molecules. What we find in DNA is the ladder or the backbone of the DNA is right-handed sugars. So all the molecules that form the backbone are all right-handed molecules, which is strange. And if you look at the amino acids that the RNA produces, they're all left-handed amino acids. And that's a real problem because in a natural probiotic soup, the molecules are either right-handed or left-handed. And to all of a sudden isolate all the right-handed sugar molecules all on one side and forcing them to join is very difficult to do without having a left-handed molecule all of a sudden be part of this. So we're going to play a little game right now. If you can turn on your video, I'm going to. So if you have a birthday, pick one person. If there's 2 of you, pick one. And if your birthday is on an odd day, put up your left hand, So if your birthday is on an odd day, you're a left-handed molecule. If your birthday is on an even day, you're on a right-handed molecule. So in order to be a right-handed molecule, we'll show everyone. Okay, let's go back here. Okay, everyone who's a right-handed molecule, put your hand up. Okay, if your birthday is on an even day, you are a right-handed molecule, which is a sugar ribose molecule. So if you look at your screen, how many people have their hands up in sequence, right? So I'm looking at this. I see 2 in a row. I see 3 in a row, 4 in a row. But so what that tells us is it's hard just amongst ourselves to have everyone chained together as a right-handed molecule. If you're looking at a simple bacteria which has 500,000 base pairs. Imagine having 500,000 right-handed people all sequentially in a row. That is very difficult to get. So as we look at amongst ourselves, just getting a right-handed people to put out their hand in a continuous sequence, I can only see maybe 4 in a row. So when people say that due to the probiotic soup, all these right-handed molecules tended to form together to create a basic DNA strand, that is quite difficult to do, and they haven't quite figured out how to do that yet. Now I have lost my. Okay, going back. Yeah, you can put your hands down. Let me go back to sharing my screen. Okay. Yeah, so as I said before, just to have simple bacteria, you need almost 500,000 of these right-handed sugar molecules. In humans, you need 3 billion, all in sequence. And yeah, it's just amazing that it actually happened. Next, we're going to go quickly into DNA storage and archiving. This is actually quite interesting if you've not know about this. So think about all the data in the world. So look at all the YouTube videos, Facebook, Google, Netflix, World Wide Web, genomic research with all, the genetic codes, Twitter, WhatsApp. If you were to take all the data in the world, it comes out to equal to about 6,000 CDs per person. And that's how much data is stored on networks and hard drives everywhere else. What is interesting is if you take the data stored within your DNA, and you've got 10 to 40 trillion cells, the information stored in your body equals the amount of data in the world. And as of 2020, the world's data comes up to about 40 to 60 zettabytes. That's 60 with 21 zeros after that. The human body is calculated to store 60 zettabytes of data. So if you consider, if you think yourself quite lowly, just remember that the amount of data stored in your body is equivalent to all the data stored in the entire world. And that's just amazing. The other thing is data archiving. So some of you have probably your wedding tapes done on videotape, probably have some data stored on CDs and DVDs. Sometimes you have some of your data stored on flash storage. Might be a good reminder to, if you have some of your cherished stuff stored on magnetic tape, might be a good idea to transfer it over, or if you have stuff on CDs or flash storage, be good to make some backup copies, because data storage has a certain life. So why am I saying this? Well, let's look at DNA. How does DNA, the data stored in our DNA, how does that compare in terms of storage 20 years, 25 years versus DVDs? Well, what they found is if you were to take your DNA and encase it in glass and throw it in a cardboard box in your garage, that DNA can last anywhere from 20,000 to 1,000,000 years. That's why when you see, I guess Jurassic Park, how a blood-sucking mosquito was encased in amber, it is theoretically possible to store the data in your DNA that could last, at least 1,000,000 years. So to me, just the way how our DNA is stored is amazing. And finally, we're going to look at DNA, RNA production of hemoglobin. Now, if you listen to the video carefully, you will actually know the answer to this. How many red blood cells are produced in your body per second? Okay, so this is a question. is it 24,000 red blood cells produced in your body per second? Put up your hand. Okay. Or is it 240,000 blood cells produced by your body per second? No. Okay. Is it 2.4 million? And the answer is 2.4 million. In your body right now, you're producing 2.4 million red blood cells per second. So that's, is that amazing? No, that's not amazing. Why is it not amazing? That's because inside each red blood cell, there's hemoglobin molecules. So the question is, how many hemoglobin molecules are there in each red blood cell? Are there 270,000? Or is there 2.7 million? Or is there 270 million? Okay. The answer is 270 million hemoglobin molecules per red blood cell. And if you do the math, okay, you're producing 70 million, 270 million molecules per red blood cell, and you're producing 2.4 million red blood cells per second. That means your body is producing 100,000 to 600,000 hemoglobin molecules per second. And that's what they were saying before in that video. Your body is producing hundreds of trillions of hemoglobin molecules per second. So that's, as we saw, that messenger RNA was actually churning out these protein chains of hemoglobin. Well, it's doing that 100 to 600 trillion times per second. But that's not amazing. You say to me, Lester, why is that not amazing? That's because in each hemoglobin molecule, there's over 9,000 atoms. So within that, it's actually putting together almost 3,000 carbon atoms. And even in this, long chain of hemoglobin, it's smart enough to know to embed 8 sulfur atoms and 4 iron atoms. So your body is not only producing hemoglobin, but it's also putting the sulfur and the iron atoms in the right location. So if you do the math, you got 2.4 million blood cells, red blood cells per second, 270 million hemoglobin molecules per red blood cell, over 9,000 atoms per hemoglobin. What that tells you is your body is arranging 1,000 to 9,000 quadrillion atoms per second. So if you feel tired someday, you can understand why, because your body is arranging almost 1,000 quadrillion atoms per second. And that's what I call amazing. So in conclusion, the language, the error correction, the optimization, the homochiral structure, homochiral structure, storage capability, archive stability, and the cell factory is just simply amazing. So the conclusion is DNA is information. It is a language. There are multiple copies, and it requires extreme elegant thought. I'm reminded of a verse in Psalm 139. It says, "I praise you because I'm fearfully and wonderfully made. Your works are wonderful. I know that full well." And as I was looking at this, I was wondering, why is it that I am fearfully and wonderfully made? And it's like, why fearfully? Why am I fearfully made? And it reminds me of a movie I saw, King Kong. And that's where you had some people walking on the island and they see this giant footprint of King Kong, and there's, talking to each other, and I go, man, whoever made that must be fearful, right? And the same way, when I look at the DNA and how it's structured, whoever put that together is fearfully powerful and fearfully intelligent and fearfully amazing. And that is the presentation. So as I said, this presentation is available to anyone who wants to use it for your own use. And we'll open up to questions. When I do Q&A at our Vancouver chapter, it's not just based on DNA, but it could be on any question on any topic. So it's probably my favorite time.
Okay, let me ask if there are any questions to start off with. Actually, I do have one, Lester, and I'm not sure that well, let me ask it anyway. And that is, the videos that we saw, are those computer animations, or, because I don't see how they could possibly be taken through a microscope.
Oh yeah, no, for sure. They're computer animation.
Okay, all right.
Yeah, but it's amazing. When I first saw that, I thought, wow, that is amazing. And it's not just, like I say, it's not just the DNA, but the ribosomes and the whole molecular machines. There are other videos on YouTube which talks about the cell, and it's just simply amazing. Just do
well, I actually kind of raises another, and this is not so much a question, I guess, as an observation, and that is, if you noticed in the animation how all the molecules seem to rush to the right place at the right time. How do you suppose they do that? What is it that causes all those molecules to rush to the right place at the right time to make all those things happen so suddenly?
Yeah, that's amazing, especially when you consider like a 1,000 quadrillion molecules. I mean, they're pumping that together in the codon. I mean, that's just one of
Yeah.
trillion molecules that are churning out the hemoglobin.
I have my own theory. Some of you have heard it before, and that is that there's something else that the science has not discovered yet that accounts for how all those things happen at the right time and the right place, and they're all those molecules are there at the right time. I think that there's more stuff that needs to be discovered, and that the science is lax in not discovering it.
Mm-hmm. What's interesting is you think about the machinery that we just witnessed, as well as the cell membrane. That is actually all programmed from the DNA. So as it's not just DNA is creating proteins, it's actually creating cells and all the molecular machines to do that. So yeah, there's extreme intelligence that, I mean, it's like a machine. It's guided. It's a factory, but it's all mindless. There's no intelligence, but it just knows to do it, and that's the amazing thing.
I like to think that they're answering or responding to the commands of the chief, Anyway, I have a question, Lester, because I've seen this presentation before, but maybe not in such details. And it's regarding homochirality.
Mhm.
There was something I was expecting you to tell us, and it was the left-handed business, and how, I can't remember if I remember this right, so you can correct me, but how something is all left-handed,
Mhm.
but you need left and right to form something or other. Do you remember what I?
Yeah, the sugar ribose backbone of the DNA, the sides of the ladder twisted, they're all made up of right-handed sugars. However, the proteins that are created from the RNA, they're all left-handed protein molecules. So that's very odd to have a left-handed protein amino acids. Yeah, they're left-handed amino acids, and amino acids come either in right-handed or left-handed. But it's kind of like a chicken and egg. How do you create a protein? Well, you need proteins in order to create the RNA, but then in order to have the proteins, they all have to be left-handed proteins, left-handed amino acids, which creates a protein chain. So in order for that to happen through chance is very difficult, which goes back to the original thing in Genesis. The Spirit of God hovered over the surface of the deep and brooded and coded and put it all together. So yeah, go ahead.
If I could comment on that, Lester, when I was taking organic chemistry, kind of in preparation for my becoming an RTB apologist, and working in the lab, I discovered that where the technology is right now is that if you are in the lab making these compounds, and we can do that, we can make all of these compounds, we can make nucleotides and amino acids and all that stuff, and we can only make what are called racemic mixtures. And a racemic mixture is 50% left-handed, 50% right-handed. We don't know how to make a one that's all left-handed or all right-handed, or even actually even a slight. We can sometimes do a slight surplus of one or the other, but we have no clue as to how to make them all left-handed or all right-handed, and yet the body does that routinely.
Yeah, unless you physically separate it right from the start. So yeah, if you were to produce it, the racemic, but you'd have to physically go in and isolate them to make them all the same, which doesn't happen in nature. You don't have a person taking it, separating them, unless you're the Spirit of God hovering over the surface of the deep.
I have a question.
Mm-hmm.
You hear in the news about, the COVID and everything else, and it's degenerating. And the question for me is that if it's degenerating or mutating, why doesn't it just, in a sense, kill itself? Because if it has some amount of mutation, which implies to me it's got some kind of replication error, that it would all of a sudden say, why does that replication error not kill the host? Because when I look at computer programming, once it makes a mistake, most of the time you get the blue screen. Mm-hmm. And so that all of a sudden you're going, but yet these viruses are, they say mutating, but yet they don't get the blue screen. At least I don't think they do. So how does that work?
My guess is this is microevolution and work. And if you have a huge population size, most of them will probably die out, but the ones that are mutant, which survive, that's how it doesn't die. There are probably a lot of strains, mutants that do die, but the ones that don't die are the Omicron and the viruses that we have now. But that's my guess. What do you think, Kirby?
Well, I think it's basically right. I mean, that there's so many millions and billions of the COVID-19 virus out there that, yeah, they're degrading, but it's only a certain percentage. Whereas, as Lester said, DNA itself can last for up to 1 million years, but RNA basically within 10 years within less than 10 years, I think RNA is pretty much gone. But we're not talking about 10 years. We're talking about, we've been going for 2 years now, and it doesn't seem like there's any end to it. But it's not as stable as DNA by any means, but it's stable enough to cause a lot of problems.
I'm just going to wade in thinking out loud here. Could it
You're a
be
nurse.
also...
You can.
Well, no, I really don't know very much about this, honestly. It's not false humility, but I couldn't help wondering if it could be because the ones that are viable have found a host, us, and because talking about the Omicron, it's so transmissible and there's so many of us. So it's sort of like found a home. Do you know what I mean? Like it's found a home, and there are lots of homes around, so it just goes from one to another. Therefore, instead of what was that, the beach ball thing or the whatever spinning, what it doesn't do that because it found a home. It found homes.
I think there's something to that. I guess we've heard that, well, maybe it came from bats, maybe it came from pangolins, but not every organism is a host for the COVID vaccine. But unfortunately, humans are a good host, looks like. So
Yeah.
Is that kind of what you're saying, Esther?
Yeah, and that's why it didn't die out as Bill was suggesting.
Right.
Yeah, not good.
Yeah.
Well, I don't want to shut off the conversation if there's more conversation going, but you people aren't asking enough questions. You're bored.
Well,
You're not bored. I know you're not bored. You're kind of, wow.
Samina's got a whole list of questions.
I have at least 3, and I'm trying to decide which one to bring up so I don't take up all the time. Well, I guess one of my questions, Lester, thank you for a fascinating presentation. And yeah, I too kept saying, wow, that's amazing. Right before you would put up the screen, I would say, that's amazing. And now I have a good thing to tell Kirk when I'm tired. My hemoglobin is working really hard. Anyway, so basically, at one point you had, and I have not taken chemistry, or well, never mind.
Biology.
Well, I might have been in biology. I can't remember. Anyway, but basically at one point you were showing the right side and you were moving the slot over a little bit, and then the left side. And I didn't understand what the significance of that was, unless your point was that even when you move it over a little, it creates different proteins, but it still is a protein. It still makes sense. It's not gobbledygook. Is that the point?
That's exactly the point. So you would have this stretch of DNA, and normally when we code as a computer programmer, basically that code just gets read once, right? But similar to a palindrome, you can read it the other way and it still makes sense. And that's the same thing with DNA. You can read it this way, it means something. You could read it backwards, the other direction, it means the same thing. But not only do you read it forward and backwards, but you could read it 3 times going this way, and then you can also read it 3 times going the other way. So the same strand of DNA, you could read 6 different ways, and it codes for 6 possible viable proteins. It's hard to do a palindrome, to read one way and back way, but to read it 6 different ways by just shifting the reading frame over is phenomenal. I mean, I don't think even the most no one can code like that. It's and
I want to point out one thing though, Lester, which I think may have been confusing to some people, and that is if you take a particular gene, there's really only one way you can code it with that DNA sequence. If you move it over even one nucleotide, you have what's called a frame error, and that will, as you implied at the beginning, that completely throws off the entire gene sequence. So it's really only good for one gene. But yeah, you can get up to 6 different genes using the same code.
Yeah, I think what's interesting is there's something called a MET, which is a start. And if you have that certain 3 combination, then it tells the transcription start coding from here.
Right.
So that's what makes the difference, is the stop and the start is it's actually embedded along the DNA. So yes, you need the stop and start. It's not like you just read any strand, the 6 ways, you need the start codon and the stop codon.
Right.
Oh, no, now I have a question. Can I ask?
Sure.
Okay, so from the diagram you show on the start and stop, it seems like they just get into the sequence randomly. You know, usually if you want to start transcription and stop transcription, you got to have a design, when to start, when to stop. And then, so how does it know where to put the start and where to put the stop?
The programmer, whoever programmed it, put it in there. It's
Right.
Yes, okay, so, but then when it's duplicating itself and then
Yep.
going through all of these different factory production and so on and so forth, how does the stop and start get put in the right place?
I believe when it comes to DNA replication, it replicates the entire DNA all in one go.
Right.
When it comes to RNA transcription, that's when it reads the start codon and the stop codon. So it only goes along the DNA, and then once it hits start, then it says, okay, start producing your protein chain.
Okay.
Now, I'm not an expert. Kirby obviously has
So-
studied.
Yeah, I think you nailed it pretty well.
Mhm. So there is way, so usually the start and stop is at the beginning and end of your DNA strand?
No,
No?
it's the start. It's a 3-letter codon, so there's only one codon that codes for start, and then there's 3 codons that's code for stop. So it's that start could be anywhere in the middle of the DNA strand, and the stop could be anywhere as well. So that stop and start is not. It has to do with the. It tells when to transcribe the DNA into a RNA strand.
Okay.
Well, it reminds me of a very common code that you'll find in every computer, and that's called ASCII. And the ASCII code also has start codes and stop codes, although they actually, I don't think they're as necessary now today as they were years and years ago, but you would send the ASCII code out over radio wires, and you had to have some way to synchronize your send and receive channels, and it was these start and stop codes that were necessary. But it's the exact same thing in DNA that does that, and that's amazing.
Yeah, it is. So help me out just a little bit more, because I'm kind of ignorant in this area. So if you got a DNA going through this ribosome or whatever machinery, right, that's replicating it, it goes through that, but there will be no replication of that DNA into an RNA until it hits the start codon, right? Then it starts.
No, because your messenger RNA. Well, yeah, I mean, your messenger RNA begins with this MET code.
Okay, it begins with the start code,
Yep.
right?
I believe
Yeah.
so. Yeah.
Yeah, okay, so it's DNA transcribed into RNA. Okay, so I'm talking about DNA going through this factory code machinery, and you pull it out, and nothing happens until a start codon is hit, and then it starts producing the RNA. Okay, and then it will keep on producing it until it hits a stop codon, and then it will stop.
Yes.
Yeah.
Okay, now, so it will produce a particular kind of RNA. But if the factory, I mean, if this machine reading mechanism reads the DNA one step over, you will get a different message, right?
Yes.
And so the start codon may be hit at a different sequence.
Yep.
Than the first time through.
Mhm.
So the factory would determine which way to read it. Is it going to read it, like method 1, or is it going to, or sequence 1, or is it going to read it sequence 2, or is it going to read it sequence 3? So
Yeah.
somehow it's got to know which way to read it, right?
Yeah, there's a book by it's called by Fuzz Rana. It's called Fit for a Purpose, and he talks about error correction within the DNA. There's something about the DNA strands that if you put a current through it, it works and it doesn't. So there's what I gave you was a high level overview.
Yeah.
There is actually error correction within the structure of the RNA and the DNA.
Yeah.
So if something doesn't get produced properly, it just won't function. So that's another level of error correction.
And John, I was going to point out that 3-letter start codon,
Mhm.
if you're reading it off, you're not going to get the same 3 letters, so you're not going to read it as a start codon.
Right.
You have to be on sequence to recognize it as a start.
Exactly.
Otherwise, it
That's
won't
what
be
I
a start.
meant. Right. So
Yeah.
so depending how the replicator is reading it, is it going to reading it by method 1 or sequence 1 or read it by sequence 2,
Yep.
And so on and so forth.
I think it's probably just looking until it sees that 3-letter sequence, and then it says, "Oh, here's the start," and then
That's
it starts going every 3 from there.
Well, it is looking 3 letters at a time. So it's, and it is possible to get it off. And if it's off, then it's off.
Yeah, the critical one is if you miss the stop, if the start codon is corrupted, then you're hooped because you're not going to get anything.
Right.
But if you have one of the other letters codons off, then chances are you will still code for the proper amino acid because there's redundancy. The only place where there isn't redundancy is the start codon because there's only one of them.
Right.
One of 64.
I was a little surprised, Lester, that you didn't mention that the error occurrence in DNA, I think it's in DNA replication, is like 1 in 10 to the 9th.
Oh, actually, that's new. I've never heard that one.
Yeah, that's amazing too.
1 in
10 to the 9th.
That's a big number. Yeah.
I had
One in a billion.
Yeah.
Okay, so I have another question, if no one else has one. This again is for my own education. So Lester, you showed a graph of a normal distribution of the replication.
Yeah, a million random DNA.
Yeah, right.
Of the genetic code. Well, of different genetic codes.
Yeah, different. And then you showed way on this side the replication accuracy of the human genome. So this one you showed is that a non-human?
No, that's just a million random computer generated codes. So,
I see.
So what they did is they just told the computer, write me 6 billion random letters.
Okay.
And we will analyze it for error redundancy.
Okay.
And from there, they kind of ranked, what is the average error redundancy? And then they came up with a normal distribution.
I got it. So the point of that curve or that graph is to show that the human replication process is so much more accurate than the man-made machine replication process.
No, it's the code.
The code is so much more accurate.
It's not just humans. It's the genetic code is the genetic code, whether it's humans or any other organism.
Oh, so okay, it's the biological.
Yeah.
Replication. Now, does that include the product after it's gone through its internal error correction, or is it without going through its error correction?
I think what they've done is they probably looked at the code and I believe they might have changed a few letters and see how well it codes to create the same protein, and then they've done that and see the reason why they did this is they thought, well, our human genetic code is just due to random processes. It's all random. There's no intelligence behind it.
Right.
But when they compare that to a million random generated codes, they find that our DNA is nowhere near the normal distribution. It's 6 standard deviations away from a random generated codes, which tells us that there's supreme intelligence required to code up our DNA.
Got it. That's amazing.
It's not due to chance. If it was done if our human DNA was due to chance, our human DNA would be somewhere on that normal curve.
Okay,
But
that's a good argument. Yes, I like that.
It's way off. 6 standard deviations is amazing.
Could I ask a
Kirk,
question?
you have your hand up. Did you mean to have your hand up? You're muted, Kirk.
Oh, I think that's just his picture.
Oh,
I know.
yeah. I think
But he
Randy
doesn't have to have his hand up, I don't think.
I think Randy had a question.
Yeah, I had a question. If there's
I guess he's gone. Sorry. Go ahead.
I had a question.
Go ahead, Randy.
Lester, I don't know if you've read Evolution 2.0, the book, but I find it just really, really interesting. It talks about, DNA from an information theory perspective, and, Claude Shannon and, and anyone who's been in telecommunications, those are all concepts that are, very, very familiar. And even he talks about, for instance, the so-called junk DNA is actually sort of contingent software. It'd be like if you bought a package of software routines and used a portion of them in an application but didn't use others, and an organism could identify at a certain point that maybe it suddenly has a use for this routine, but it's already been built in by the creator. And so the fact that a creature isn't using it at this instant in time doesn't mean that it's junk, and so, but anyway, even when creatures mate, some of that is passed on and so forth. So anyway, I was very interested in, just the whole view of basically God's providence from an information, perspective.
Mm-hmm. Actually, that's a very good point, because when we look at human DNA, right, and we realize that this DNA has to control the body from when you're a newborn to, when you're old like me, where you're getting, your body changes, right? You're going through puberty, you're going through certain phases in your life. And obviously, the DNA doesn't kick in until you hit a certain age when your body starts changing. So that DNA has a lot of data or programming that is not used until you hit a certain phase in life. So the fact that we have it in such a small little strand, 6 billion,
Oh, Lester, you're frozen.
life from,
there you go. You were frozen for a minute. I don't know how much we missed.
Oh, really? Yeah, essentially what I said is the DNA is programming our life from when we're a newborn to when we're old. So obviously with a newborn, there's certain processes happening in their body, and then you go through adolescence, puberty, and then as you get older, your body changes. So a lot of times certain codes in your body won't be used until 50 years down the road.
Mm-hmm.
Yeah, that's all stored and coded in our DNA, and in such a small, compact way.
Yeah, that's amazing. Now, so what about human consciousness and human spirit? If the more we study these DNA and replication, we're just a bunch of machines, micro machines, right? Tiny little machines built together. But then how does that it's hard to believe that translates or could translate into consciousness or human spirit. So therefore, one would think that the consciousness and spirit comes from a different angle. And this consciousness and spirit just uses the human body to derive information, to derive consciousness. Could you comment on that?
Yeah, in the very first 3 slides, we talked about your worldview has to answer these questions. One has to do with purpose in life, why we're here. Second one has to do with morality. The other one has to do with destiny. But the fourth question was the origin. How do you explain the origin of life, the origin of the universe, and the origin of consciousness? And you can't achieve consciousness from inanimate objects. You can only achieve consciousness from something that already has consciousness. And the Bible tells us that God created us in his image. So because God has intelligence and consciousness, we also have consciousness and intelligence as well. So from a Christian worldview, Christianity answers the question of consciousness in the best way compared to other worldviews. That's a simple explanation, but from the more I look at the four basic questions, Christianity comes on top and answers it in a coherent and a way that actually makes sense as well. So
yeah, it's like a robot. You know, a robot, man makes a robot, and they're getting more and more sophisticated. But a robot is just a bunch of tiny little machine modules put together to do certain things. But it doesn't have a consciousness until it's programmed to mimic a consciousness. But it doesn't have the consciousness of free will like we do. It can only do whatever has been programmed in, and that's it. It's limited.
yeah, if you watch some scientific movies, you know, they're saying, you know, artificial intelligence should end up, you know, becoming self-aware, which may or may not. But if you realize that whoever programmed the AI required intelligence to program it that way to self-learn. So it didn't happen from nothing, in the same way that for us to achieve self-consciousness, either through AI or self-learning, it had to be programmed in a way to make it happen. So you still can't get away from God. You still need an intelligent programmer to make it happen. I'm not sure if it is possible to have self-consciousness in a program, but I can't remember whether Fuzz says it's contrary to what you see in the movies and on Star Trek, and we're quite a far distance away from that.
Well, if there are no further questions, I think I'm going to arbitrarily and capriciously end this meeting. Okay.
Question.
Go ahead, Bob.
These have nothing to do with the content.
Okay, yeah.
These are simple. Number one, I have a friend who wants to see this presentation. Is it available anywhere? And number two is I've got my own record button down here. I've never recorded anything from a Zoom meeting, but if I did record it in the future, would I be violating anybody's rights or space or desires or anything like that?
Not mine.
No, that's fine. I actually checked with RTB, and there's nothing here that's copyright. So
for those of you who didn't hear it, Lester offered to send out his PowerPoints, as well as I am recording this lecture, and I will forward it to all of you, as well as the slides. So if that's what you're asking for, consider it done. Well, it will be done. It hasn't been done yet.
Great. That's great.
yeah, although the cartoons have copyright on it, I got my friend to do it, and he sold me the copyright for free. So even though it's copyright, it's not. It's free for any use amongst the RTB people.
Well, and you said it's on YouTube. I mean, we can.
Oh no, the visualizations are on YouTube, but
Right.
the actual slides itself, those are.
Oh yeah. Okay.
The actual slides with the pictures are. Yeah, you could do whatever you want with it. You could copy it, give it away. I would hope that you would reuse it to show in your Sunday school class or anywhere to promote the idea that we are fearfully made, and that of all the worldviews, only the Christian worldview makes sense of the origin of life.
Let me close this meeting with a word of prayer, and I want to thank you, Lester, for for having done this this lecture. I think everybody enjoyed it, and let's bow for a closing prayer. Gracious Lord God, thank you so much for your amazing DNA, for what you how you have created us. We are definitely fearfully and wonderfully made, and we thank you so much, and that you have created us in your image, and it's just we're constantly blown away by what we see, and we just thank you, Lord, and ask that you would go with us now as we continue our lives, and we just praise you, and we just ask for your blessing on us, each one. Thank you. We pray these things in Jesus' name. Amen.
Amen.